ดรยันบินเฉินนะครับGlobal Technical Service Director ของบริษัท APC นะครับจะมาคุยเรื่องเกี่ยวกับการสร้างเสริมภูมิคุ้มกันต่อในตัวสัตว์ให้ทนต่อให้สามารถผ่านโรควิกฤตโรคนี้ไปได้นะครับดรยันบินเฉินเองเป็นทีมงานของเรานะครับที่ทำเกี่ยวกับเรื่องการรีเสิร์ชหรือทดลองเรื่อง develop การใช้ในอาหารสัตว์นะครับโดยเฉพาะในสุกรนะครับแล้วก็เคยเป็นทีมงานตำแหน่งเก่าเธอทำงานอยู่ที่บริษัทคาร์กิลที่อเมริกาด้วยนะครับ
Thank you, Waraz, for the introduction, and thank you all the participants for getting on the call with us today, and thank you for your time. Today, what I would like to talk to you about is about the plasma and the African swine fever virus. As you all know, since African swine fever virus being introduced to China in 2018, and the virus has quickly spread all around Asia. It has caused tremendous loss in all the countries it has entered, and also it became the number one threat to the global swine industry. So we really need to understand this virus and also understand what are the potential tools that are available to help us to deal with this virus. And today I would like to share with you a study we did in Spain just last year on looking at the role of dietary spray-dried porcine plasma on the pigs that are being challenged with African swine fever virus. To fight your enemy, you have to really fully understand your enemy first, right? So to fight against the African swine fever virus, we first need to understand fully what are so unique about the African swine fever virus that may give us the edge to deal with it. Here I'm listing two really unique features about this virus that really can potentially help us to win the battle. The first feature about the African swine fever virus is the host cells of African swine fever virus. So from the publications and the literature, we know this virus only infects macrophage cells and the dendritic cells of a pig. Those are immune cells. And this virus does not infect epithelial cells. For example, the epithelial cells in gut or lung or skins, those are not the host cells. What it means is the virus has to penetrate through this epithelial layer to get in contact to its host cell, which are the macrophage and dendritic cells. So this is meaning that if we can somehow protect the epithelial layer healthy, make sure it's intact, that there's no leakage and there's no damage of the epithelial layers, we can potentially help the pigs to reduce infections or at least increase the minimum infectious dose by this virus. So this is very important. I'm going to show you a slide later on to fully explain to you what does this mean. Number two feature of this virus is the pigs, when it get infected, the pigs does not seem to be able to produce neutralizing antibodies. That's one of the reasons why this virus has so high mortality rate, because the antibodies, they don't have neutralizing antibodies to fight against this virus. However, we do know from the paper and also from the field experiences in China or other countries, that the serum, the covalent serum from infected or recovered pigs, if you use that serum injected to the other pigs, it will help to increase the survivability of those other pigs. So there is something in the serum, not the antibody, but there is something in the serum can potentially help a pig to survive from this horrible challenge. So those are the two unique features of African swine fever virus. Move on, just to fully explain to you the first point I made, which are the importance of the epithelial layer health and the risk of virus infection, especially for African swine fever virus. Here, you're seeing a picture of the gut epithelial layers, right? You have the gut lumen, you have all the nutrient, all the feed, and all the microbe or viruses on the lumen. And then you have your epithelial layers, and under the epithelial layers, it's what they call the pneumonia and propria layer. So that's where the immune cells are located, which, in the purple, those are macrophage cells. And this in blue, those are dendritic cells. So they actually live under the epithelial layers in this luminal purpureal portion of the gut epithelial. So, in a normal condition, a very healthy pig has a very healthy epithelial layer. They're tight and there's no leakage. And the viruses or the microbe, they have very little chance to penetrate through. And therefore, for example, for African swine fever virus, it has little chance to penetrate through. It has little chance to meet its host cell, which again are macrophage cells and the dendritic cells. And that means it will not cause infections. However, in a stressed condition or in a diarrhea condition, when the gut has been damaged, and then that means the bacteria or the viruses from the lumen can penetrate into the pneumonia propria portion of the epithelial, where the African swine fever virus can meet its host cell, again, our macrophage cells and dendritic cells, and cause infection. So here you can see, the epithelial layer health and whether there's leakage or not, or there's damage or not, are very important, especially for African swine fever virus infection through gut. Those are the kind of the theory. This is a paper really proved that theory. This is a paper published by a German scientist, Sandra Blum, from her lab. She really are an expert in African swine fever virus. This is a paper published in 2015, where she looked at through the oral nasal administration. She looked at whether healthy pig or unhealthy pig has a different minimum infection dose. So basically, what she found out is when she gave the virus, a live virus, through oral nasal way at 100 unit per pig, she could not infect the healthy pigs and that are in this trial. And however, when she gave the same way of administration, but the lower dose, 10 unit, she was able to infect in this particular trial an immunocompromised run pigs in her study. So apparently, those immunocompromised run pigs, which are all having this gastric intestinal infection and diarrheas, that all have a lower infection dose needed to get them infected with African swine fever virus. So in conclusion, this study concluded that herd health, the health status of the pig, especially the mucosa health and integrity, and also the immune response at the mucosa layer are keys for African swine fever infection and African swine fever virus control. So, back to the slides, we now know to maintain this epithelial layer health, it's very, very important, especially for controlling African swine fever virus infection. Now the question is, how do we maintain it healthier, right? Plasma has long been studied and showing that it can improve the epithelial layer health. It can improve the tight junction between the cells, improve the barrier function, reduce inflammation. And also there's another unique feature of plasma, which actually can help those damaged epithelial layer to repair to the normal stage, because there are some growth factors in the plasma that can promote this process really to help the damaged layer coming back to the normal stage much quicker compared to other feed ingredient. So this is important. And also in plasma, there are components in plasma have this immune regulation properties, which we believe may have contributed to the health benefit from plasma, right? Plasma has been long been used for example, for controlling PRRS viruses or controlling other bacterial infections like E. coli or Salmonella infections in pigs. We believe those are coming from those specific component from the plasma. For example, immunoglobulins, the IgGs, right? They have a neutralizing capability that can neutralize pathogens and facilitate immune response, help pigs to get back to their normal immune stage quicker. And also there are some bioactive proteins in plasma has these antiviral activities, like interferon gammas or alphas. They actually can inhibit the virus replication, also facilitate the proper immune response. Here, I'm calling out the CD8 T cell response. This is very important for actually controlling African swine fever virus. But there are some component in the plasma, as I showed you before, may have a role to contribute to the control of the virus in the pig. Here are two studies really showing the components of the proteins in plasma. The functional proteins actually has the ability to inhibit virus in replication. On the left side, those are the study from years ago, very common sense for biologists, which is the fetal bovine serum has ability to inhibit flu virus replication, and they discovered this by when they are doing this flu virus or flu vaccine development, right? When they are incubating the vaccine, these viruses, and they have to pull out the bovine serum because the serum has a really strong impact on the flu virus replication. We know the serum, bovine serum, fetal bovine serum, because they're from the fetus, it doesn't have any neutralizing antibodies against the flu virus, right? So there is something else in the serum are inhibiting the virus, which we believe is those bioactive components like interferon gammas or alphas. Those proteins are inhibiting the virus replication. So on the left side, as you can see, the fetal bovine serum has inhibition rate of the viruses around 100,000 times. So if you have a bovine serum, and you can reduce the replication rate by 100,000 times. On the right side, it's a new study just published last year by Harbin Institute of Veterinary Research. They really specialize in African swine fever virus research in China, where they published the data showing the interferons from pork blood can really inhibit African swine fever virus replication. And the conclusion is those interferons can reduce the replication by over 100 times, in pigs and also in the cell culture setting. So again, this is showing you the functional proteins not only are providing, the nutritional effect, but also are providing the functional effect that have a profound impact on the health of animals, especially if the animal are challenged with either bacteria or viruses. So with that in mind, we actually run the African swine fever virus research in Spain, in CRASA, which are a federal facility from Spain. They're specialized in African swine fever virus research. The goal of this study is trying to look at whether to characterize by feeding both spray-dried porcine plasma to pigs that are being challenged or injected with African swine fever virus, how would that pigs respond? And the second objective is to determine whether this feeding of spray-dried plasma can actually reduce the transmission rate of African swine fever virus, between pigs in the same pen, because we hypothesize the plasma, by feeding plasma, it might have a health impact of the pigs, and also it might have the impact of the virus replication in those injected the pig, may reduce the viral spreading, therefore reduce the transmission, in the same pen or in a barns. So this is a study we run. Put this away. This is a study we run. We have two groups. Group A, we were feeding them 8% plasma. On top, 8% plasma inclusion in the diet. Group B, we're feeding them a controlled diet, corn, soy, controlled diet. And those are the two groups. Each group had 12 pigs. They were hosted in biosecurity level 3 virology lab in two different rooms. So each group have 12 pigs. The pigs we're collecting are healthy, naive pigs, and they have never been exposed to African swine fever virus before, so they are clean pigs. We brought them into the lab and put them into the pens, give them about 4 days to accommodate to the pens. Then on the experimental day 0, we selected 2 pigs from each group, so a total of 4 pigs. Two pigs from each group. We injected those 2 pigs with live virus, African swine fever virus, the Georgia strain, at the dose of 10 to the 3 unit. We injected those pigs and have those pigs infected artificially. Then we follow those 2 pigs for 2 days, and we confirmed that those 2 pigs actually really are infected. Then we put those 2 pigs, back to their original pen. So 2 pigs, 2 infected pigs back into the group A pens, and 2 infected pigs back to the group B pens as well. So then we follow those 2 pigs and also the pigs in the same pen for another 4 days. On day 6, we found out that all the Trojan pigs, the injected pig, they are all having high fever, including group A and group B. Those 4 pigs are all having high fevers. Due to the experimental protocol, the lab policies, if those pig has consistent high fever, we have to kill them because of the welfare concerns. So we euthanized all 4 pigs, 2 pigs from group A, 2 pigs from group B on day 6. Then we followed all the contact pigs in the same pen from group A and group B all the way to day 19. In the meanwhile, we collected samples from them to try to determine whether those contact pigs from different group, whether more or less of the pig are getting infected or not, right? The goal is to show the dietary impact on the transmission rate, which is, the percentage of the pigs in the contact pigs population to get infected or not. However, unfortunately, on day 18, we find out that actually all the contact pigs, either group A or group B, all of them are clean. They were all not. None of the pigs were infected by African swine fever virus. We figured probably because we killed those Trojan pigs too early at day 6, and we probably didn't give them enough viruses in their pens to cause infection. So then we decided, we need to challenge them again. So on day 19, from the remaining pigs, we took another. So we started with 12 pigs. We euthanized 2. Now we have 10, right? From the remaining 10 pigs, we took out another 3 pigs, and again we injected them with the same virus and with same dose. But this time, instead of taking them out when they have high fever, we decided to keep them in the pen until they naturally died in the pen, really to give more virus threading in the pen to challenge those contact pigs. Then we follow the pigs all the way to day 31. Then we also euthanize all the pigs at the day 31 and collect all the samples. So this is the timeline and how we conducted the trial. Let's look at the result. Here's the result on the temperature. For every pig in this study, we tracked their body temperature. On the top, you're looking at is the plasma group, all the pigs' temperature over the experimental period. On the bottom, you're looking at the temperature of the control group. As you can see on the left side, those two pigs, the red line over here, those are the first two children after we gave them the injection on day 0, and they quickly became, are having high fever. On day 6, we euthanize them. Similarly to the control pig, we euthanize them on day 6. On the right side, as you can see, those are again another 3 red line. The 3 red line here in the plasma group, those are the second group of children. We injected them on day 19. All of them are starting having high fever, but they picked on day 24, 25. Then they start to normalize and start to temperature start to coming down for the children pig. In the contrary, for the control pig, after we injected them with virus on day 19, all of them are starting to have high fever, and all of the pig all the 3 pigs died within 4 to 7 days after injection. And for the plasma group, I didn't mention 2 of 3 injected pig actually survived until the end of study, versus the control pig, all 3 pigs died quickly after injection. So they naturally died quickly. Those are the children pig. Let's compare the contact pig. On the top again, if you can look at those colored lines, those are the contact pigs. As you can see, their temperature really stayed very, very low until really at the end of the study, where some of the pigs are showing high fever. Versus the control pig, as you can see, all of them are starting to have high fever very early on, in the study, day 25 or 26. So the onset of fever is much quicker in the control group compared to the plasma group. Again, this is the average temperature, excluding those children pigs of two group. As you can see, on the yellow line, those are the control group pigs, and the blue line is the plasma pigs. As you can see, the control pigs, they really develop high fever much quicker, and the temperature is much higher compared to the plasma group. This is indicating that the infection probably happened much quicker in the control pigs than in the plasma pigs. This is another view of all the pigs, and try to show you how they behave each individually in this trial. As you can see on the left side, it is all the pigs in the plasma group, and then each box over here are actually a day, right? If the box is green, that means that pigs in that experimental days, they are having normal temperature. If they became red, that means they're showing a high fever. As you can see, we have two first round children, 2 second round children, and the contact pigs. The 2 first round children, we give them the injection on day 0, they develop high fever, and on day 6, we euthanize them. The second round children in the plasma group, we give an injection on day 19, and they develop high fever. 1 pig naturally died at day 26, but 2 of them actually survived until day 31. This is very, very unusual because this is the Georgia strain. It is reportedly that the mortality rate for this strain is 100%. In the facility where we're running the experiment in Spain, the mortality rate is 100%. So there's and the professor was very surprised that those two pigs, after receiving plasma, those two pigs actually survived until day 31. They didn't die. And the control pigs in the plasma group, as you can see, some of them didn't really show any high fever at the end of the study. Even at the end of the study, some of them are developing some fevers at the end of the study. If we compare to the control group, again, the first two Trojans, they are behaving very similarly, but the second two Trojan, 3 of them all died very, very quickly compared to the plasma group. Only 1 died. And also the contact pig. The contact pig are having much higher fever at much earlier phase of the study, right? And they're having high fever starting from day 27 to 29. Also, in this study, we collected blood samples, nostril swab, rectal swab, and also tissue samples from all the pigs. The idea is really try to identify the viral load in those pigs. And this slide really shows you the result of the viral load in blood and nostril swab or rectal swab of the Trojan pigs at the day 6 post-injection. On top, you have plasma group. On the bottom, you have the control group. Here, you're looking at the CT values. The CT value is basically a PCR test of the virus. The higher the value is, the lower DNA copy there is. So the higher the value means the lower the viral load is. As you can see, the plasma group on average in blood and nostril swab and rectal swab compared to the control group all has a higher CT value, meaning they have a lower DNA copy. So for you to understand, for us to really look at it clearly, I translated this CT value using an equation to the DNA copies of the virus. As you can see, this is the number of the DNA copies in blood, nostril swab, rectal swab for the plasma group and for the control group. As you can see, there's a lot of viruses in the blood. But plasma really decreased the DNA copies in the blood sample, nostril swab, and rectal swab. And if you look at the fold changes, it's between 7.8-fold to 60-fold, almost 60-fold differences between those two groups. Really, this is showing you plasma, by feeding plasma, reduces the viral load in the blood and nostril and rectal swab of the Trojan pigs. We also swapped, the contact pig as well at the end of study, which is day 31. We did the same, same PCR test, and then did the DNA copy translation. As you can see, again, in the blood and nostril swab, the fold, plasma really decreased the DNA copies of contact pig as well, by decreasing about 140-fold in blood and 7-fold in nostril swab. In the rectal sample, the level are so low, plasma actually showed higher DNA copies. But the level is so low that the accuracy here may not be as accurate as, for example, in blood. Again, beyond the blood sample, we also collected tissue samples to make try to identify the viral load in the tissues. We know African swine fever virus mainly replicates itself in the lymph node. So we collected all the lymph node, the gastric, hepatic lymph nodes, spleen, basically all the lymph node and toe cells, and try to identify the viral load in those tissues. Again, as you can see, I'm showing you here is the mean CT values, and the blue is it's plasma. And the yellow is control group. The higher CT value is, meaning the lower the viral load is, right? And if you look at the DNA copies, the plasma level consistently, plasma group consistently had the lower viral load. the fold differences between two groups are very significant, ranging from about 200-fold differences to 3,000 differences between those two groups. This is in children pigs. Also in a contact pig, at the end of the study, we collected all the samples, all the tissue samples. It showed the same story. In all the tissues we have collected, plasma group all had a lower viral load compared to control group. So in conclusion, we can see plasma reduced the viral load in blood, nostril, and rectum swab of children pigs, and also reduced significantly viral load of contact pigs in all the tissues. We also, seem to see that the plasma may delay the infection or onset of the clinical symptom in a contact pig, probably because of the reduced viral load in the environment or threaded by the infected pigs. So that's why we see that the reduced or delayed infection. Also, we think use spray-dried porcine plasma in feed in African swine fever virus endemic region may be helpful to reduce the risk of infections by contact. So definitely, it's a tool that you might want to consider if you're facing this challenge. Of course, plasma, spray-dried plasma is a highly effective biological product, right? So we want to also show you how spray-dried plasma is made and to ensure you the safety of this biological product. This chart shows you how plasma is made. Really, it starts from blood collection. All the plasma we produced from APC, and the blood is collected from a federal inspected facility. Here we're saying USDA. This is from American inspected facility. Really, this is ensuring that all the pigs arriving at the slaughter plant that are being killed, we're collecting blood from. Those are animals that has passed antemortem inspection. Those are healthy animals. We're collecting the blood from healthy animals. Those animals are fed for slaughter for human consumption. So the blood we're getting are really clean blood. Then after we collected the blood at a slaughter plant, it's quickly been isolated and processed at a slaughter plant enclosed tank to ensure there's no cross-contamination at the slaughter plant. Then we have APC has a dedicated tanker to different plants to take the blood and transport it to our manufacturing facility. APC, we have both pork plasma and also bovine plasma. We have a dedicated tankers and dedicated manufacturing plant for either pork plasma or bovine plasma. So those are all separated and segregated to ensure that pork is 100% pork and bovine is 100% bovine. At our manufacturing plant, the plasma will be filtered and they will be processed. There are multiple steps, Q steps, built in our manufacturing process to ensure we have a full inactivations of all the potential microbe of concerns. One of them are UV treatment. The other are spray dry, and the high temperature in spray dry. It's a very effective Q step. And another one is holding the product for 14 days at the room temperature. It's another way to inactivate viruses of concern. And then all the product will be PCR tested prior to release. So as I mentioned, our manufacturing process really has 3-step built in what we call multiple hurdle technology to fully ensure that the product's been treated to its fullest extent to ensure the safety of the product. those steps are UV treatment, spray dry, and post-dry heating. And last year, we have engaged the University of Minnesota to do a risk assessment, about spray dry porcine plasma production, and also an assessment on our manufacturing process to determine the efficacies of the manufacturing process. Through that risk assessment, the University of Minnesota, the third-party university, concluded that by using spray dry, post-dry heating plus UV inactivation, would have sufficient inactivations of all the viruses under all calculated scenarios. So basically, those three building steps will help us to mitigate all the risk perceived, or inactivate all the viruses of concern. This slides, this is a new publication just published this list last month by European regulatory agencies. EFSA, European Food Safety Agencies, they did a risk assessment of all the feed ingredients that be used in their regions. And the scope of the study is trying to look at what are the ingredients may pose a higher risk of introducing, for example, African swine fever virus to EU regions, right? EU has African swine fever virus, so that's why they're looking at this, feed ingredient, which one has a higher risk, which one has a lower risk. Essentially, what they concluded is the hydrolyzed protein and also spray-dried blood product ranked the lowest on that list. Basically, they have the lowest risk associated with them for introducing African swine fever virus. On the list, you have feed additive, mash feed, and pellet feed or cereals, and those have a higher or much higher risk profile than, for example, hydrolyzed protein or blood product. This is very interesting, and basically, in line with University of Minnesota's conclusion, which is because blood products are being highly regulated, and also we have that built-in kill steps in our manufacturing process, and that's why the blood product has very low risk of introducing, for example, African swine fever virus in Europe. But this is evaluation trees or decision trees. A lot of veterinarians tend to use this to determine the risk profile of a feed ingredient. The first question they will ask is the ingredient manufactured or packaged in a country that have active viral transmission or no? If it is no, then there's low risk. APC products are all manufactured from African swine fever virus free region or country. So the answer for that question is no. That means all the APC product has basically zero risk of transmitting African swine fever virus. And also APC products, as I showed you, are all manufactured with validated mitigation process that are efficient to inactivate all the viruses of concern. So therefore, there's another safety guarantee on top of, we're manufactured from African swine fever free country. So the product we're supplying to our customer are very safe product. In conclusion, what we learned is plasma has long been used to improve swine herd health and resilience to disease challenge. And we have also showed you plasma reduced African swine fever virus viral load in blood and also virus of tissues of infected pig and delayed infections of contact pigs. We believe dietary strategy that improve gut health and directly impact immune system should be considered for African swine fever virus control. And we believe plasma protein manufactured using multiple hurdle technology are a safe and effective functional ingredient should be considered for pigs and should also be considered for African swine fever virus control. That's it. Thank you for your attention. I'm going to stop sharing. What else? Stage is yours.
Thank you, Yanbin, for your presentation. Okay. I would like to summarize for the attendee. Okay. ผมสรุปอย่างนี้นะครับขออนุญาตสรุปข้อมูลของดร.ยานบินให้นะครับคือข้อมูลที่เขาให้มาก็คือที่เราพบคืออันที่หนึ่งก็คือเราพบว่าแน่ๆ ASF เนี่ยมันจะไม่ติดเข้าผ่านทางอิพิธีเลียมเซลล์ของลำไส้แน่ๆไม่ใช่ตัวหลักนะครับแต่การติดเข้าไปจะเป็นส่วนการติดของโมโนไซต์หรือกลุ่มเม็ดเลือดขาวพวกแมคโครฟาจหรือเดนไดเซลล์ ที่ทำงานนะครับโดยหลักการปัญหาที่เกิดขึ้นที่เราพบก็คือเกิดจากลำไส้เกิดการติดเชื้อหรือมีลักษณะของการเป็นกัดลีกหรือเกิดช่องว่างของลำไส้ที่เกิดความเสียหายเนื่องจากการอักเสบก็ตามแต่ทำให้เม็ดเลือดขาวทำงานเยอะขึ้นถึงเกิดการติดเชื้อ ASF ได้ง่ายขึ้นนั่นเองนะครับแล้วก็จากการข้อมูลที่เขาได้มาเพราะว่าสุกรที่สุขภาพดีจะมีโอกาสในการติดเชื้อน้อยกว่าสุกรที่สุขภาพไม่ดีนะครับหมายความว่าถ้าสัตว์เฮลตี้ดีโอกาสเสี่ยงหรือโอกาสที่จะติดเชื้อก็น้อยลงไปด้วยนะครับดังนั้นแล้วความสำคัญในการดูแลสุกรแล้วก็คือทำให้สุกรมีสุขภาพที่ดีและแข็งแรงนะครับเพราะปริมาณเชื้อในการติดเนี่ยของสุกรสุขภาพไม่ดีเนี่ยใช้นิดหน่อยก็ติดแล้วนะครับอันต่อไปก็คือส่วนสำคัญในการป้องกันการติดเชื้อนี่ก็คือเรื่องของสุขภาพของลำไส้เองนะครับส่วนของ intestinal barrier คือผนังลำไส้เองต้องแข็งแรงนะครับไม่มีการลอกหลุดของตัวเยื่อบุอิพิธีเลียมทำให้ไวรัสไม่สามารถผ่านเข้าไปได้นะครับmucosal immunity หรือ mucosal ที่ครอบคลุมก็เป็นตัวหนึ่งที่ช่วยได้นะครับหลักการลดการที่ดีคือการลดการอักเสบของลำไส้ทำให้สัตว์สามารถกินอาหารย่อยได้ดูดซึมได้แล้วก็ไม่มีปัญหาเรื่องการติดเชื้อนะครับส่วนในการทดลองเขาที่ทำเกี่ยวกับ ASF นั้นโดยใช้พลาสมาโปรตีนที่ 8% ในอาหารแล้วเทียบกับกลุ่มคอนโทรลนะครับแล้วก็พบว่าสิ่งที่ได้ก็คือเราจะเห็นภาพของสัตว์ที่ติดเชื้อที่เราใส่เข้าไปเนี่ยในช่วงแรกเนี่ยเนื่องจากข้อกำหนดของสวัสดิภาพสัตว์ทำให้เราไม่สามารถคอนตินิวการทดลองได้นะครับหลังจากเข้าไปได้ 3-4 วันเราก็ต้องเอาออกมาเนื่องจากสัตว์มีไข้สูงแล้วก็ต้องทำลายทิ้งนะครับแต่เราพบว่าในช่วงแรกที่ใส่ไปในกลุ่มแรกการทดลองเนี่ยไม่พบการเฉียดเชื้อในส่วนของผมเองผมมองว่าอันนี้เป็นข้อดีอย่างนี้เราเห็นภาพว่าถ้าเราสามารถค้นพบตัวสัตว์ที่ป่วยได้เร็วเราสามารถคัดออกได้เร็วปริมาณการเกิดการติดเชื้อในฟาร์มก็จะลดความเสี่ยงไปได้มากขึ้นนะครับส่วนช่วงที่2ที่เขามีการใส่เพิ่มเข้าไปอีกนะครับในส่วนการเอาใช้กลุ่มเดิมนั่นแหละที่ผ่านการกินพลาสมาแล้วและไม่ได้กินเนี่ยเอามาแล้วก็ทำให้ติดเชื้อแล้วใส่กลับเข้าไปแล้วปล่อยจนกระทั่งผู้นั้นอยู่ในฝูงจนกระทั่งถึงตายนะครับจนเสียชีวิตไปนั่นเองนะครับแล้วเพราะว่าในกลุ่มที่กินพลาสมาเนี่ยมีสุกรตายไปแค่ตัวเดียวที่เหลือยังคงมีชีวิตดำรงอยู่ต่อไปได้จนถึงสิ้นสุดการทดลองแล้วเราเอามาผ่าซากพิสูจน์ดูว่าเกิดอะไรขึ้นแล้วก็เช็คตรวจเลือดนะครับการในกลุ่มที่คอนโทรลเนี่ยเพราะว่าเกิด3ตัวที่ติดเชื้อมาเนี่ยเสียชีวิตทั้งหมดหรือตายทั้งหมดนั่นเองเป็นตัวบอกว่าพลาสมาน่าจะมีคุณสมบัติบางอย่างที่ช่วยให้ประคับประคองตัวสัตว์ให้สภาพที่ดีและลดการติดเชื้อได้ซึ่งผลจากการตรวจเลือดนะครับการสวอปจากจมูกแล้วก็สวอปจากก้นนะครับหรือฟีคอลสวอปเนี่ยเราพบว่าไวรัสโหลดในกลุ่มที่กินพลาสมาต่ำกว่ากลุ่มที่ไม่ได้กินพลาสมานั่นเป็นตัวบ่งชี้ว่าพลาสมาน่าจะช่วยเรื่องการลดปริมาณไวรัสในตัวสัตว์ทำให้เปอร์เซ็นต์ในการปล่อยเชื้อหรือเฉียดเชื้อลดลงไปด้วยนะครับซึ่งเป็นข้อมูลที่น่าสนใจนะครับที่เอามานำเสนอในวันนี้นะครับ